This video provides evidence that mRub0173 is orthologous to E. coli serA, mRub0125 is orthologous to E. coli serB, and mRub2910 is orthologous to E. coli glyA. Now, mRuber is a gram-negative bacteria. It's rod-shaped and has a red pigment. It's non-motile and non-spore-forming, and it's a thermophilic bacteria that grows at temperatures ranging 35 to 70 degrees Celsius. It was originally isolated in the hot springs of Russia, but more recently it's been a contaminant in paper mill pulp where it creates a biofilm. Why would we want to study mRuber genes? Well, it comes from a relatively understudied branch in the tree of life. Dr. Scott chose to study the lesser-known organism because it may also lead to novel processes that we haven't found in more well-studied bacteria. Serine and glycine are well-studied amino acids. They are 2 of the 20 basic amino acids. Serine, as you can see in the top right here, is a non-essential protein that is functionally present in many proteins. Glycine is here on the lower right-hand corner of the screen. It has a unique hydrogen side chain that increases rigidity and conformation stability. Because of this increased stability, there's an increased concentration of glycine in thermophilic proteins. Both glycine and serine are needed for cell growth and play a big part in the production of DNA. In particular, glycine is more interesting to us because mRuber is a thermophilic bacterium. For serine biosynthesis, there are 3 steps in the pathway, and they'll have enzymes. The pathway is from 3-phosphodeglycerate to L-serine. The 2 enzymes we're focused on are D3-phosphoglycerate dehydrogenase, that's encoded by serA in E. coli, and phosphoserine phosphatase that is encoded by serB in E. coli. serA catalyzes the reaction of 3-phosphodeglycerate to 3-phosphohydroxypyruvate. During this, it oxidizes NAD+ to NADH+. serB, on the other hand, is part of the last step in the pathway where it takes 3-phospho-L-serine and hydrolyzes it to L-serine. In this process, it takes a water molecule, and phosphate is a byproduct. The other gene that we are studying is a part of the glycine biosynthesis degradation one-step pathway. We're interested in the glycine hydroxymethyltransferase, also called serine hydroxymethyltransferase. It's encoded by glyA in E. coli, and it catalyzes the reversible reaction between glycine and L-serine. For our first comparison of mRub0173 and E. coli serA, we first did a NCBI blast search. The amino acid sequence of mRub0173 was the query sequence, and we did it against the amino acid sequence of E. coli serB as the subject sequence. You can see a lot of similarity where they line up. The middle one is the same as the top 2, and the pluses mean that they're both polar or they're both nonpolar. We get a very low E value of 4 to the negative 41. And even though this score is very low, we can justify it because E. coli and mRuber are not closely related species. Nevertheless, the low E value indicates a strong sequence similarity. This slide is the result of the additional bioinformatics tools that we use to compare E. coli serA and mRub0173. In addition to the strong blast alignment, they both pulled the same COG hit, which is COG 01111. They had the same TigerFam hit, which is TIGR 01327, which is phosphoglycerate dehydrogenase, and they pulled the same PFAM hits, which was the 2-acid DH and the 2-acid DHC. All of these have very low E values, which indicates strong sequence similarity between E. coli serA and mRub0173. The protein database pulled two different numbers, but as you can see, it's just phosphoglycerate dehydrogenase, and they're just in two different organisms. And we can justify this because, again, E. coli and mRub are not closely related species. On top of all this, they have the same predicted EC number, which is 11195, which is phosphoglycerate dehydrogenase, and nothing on this table indicates that mRub0173 is not phosphoglycerate dehydrogenase. In conclusion, for this comparison, there's a strong E value. The initial blast, they have been proven to be in the same cellular location, which is the cytoplasm. They both pull the same COG numbers, PFAM numbers, and TigerFAM hits from the databases. The PDB results were different, but again, they're the same enzyme in the different organisms. They're both predicted to have the same KEG map in serine biosynthesis. Both are predicted to have the same EC number. They have similar conserved amino acids when comparing PFAM conserved sequence, and neither of them are a member of an operon. For our next comparison, we're looking at mRub0125 and E. coli serB, and we once again did the initial comparison of the NCBI blast. The query sequence was mRub0125, and the subject sequence was serB. Again, we get a low E value of 8 to the 0.6. Again, the bit score was a little low, but we can again justify this because E. coli and mRub are not closely related species. Overall, the low E value indicates strong sequence similarity. This table is the rest of the results of the other bioinformatics tools that we use to compare E. coli serB and mRub0125. Again, in addition to a strong blast alignment, they pulled the same COG number, COG 0560, just serB. They did pull two different TigerFAM numbers, but that's okay because with more research we found that phosphoserine phosphatase is actually in the HAD hydrolase family, and they pulled the same PFAM number, PF02710. All of these have very low E values, which indicates sequence similarity. The protein database pulled two different numbers. E. coli serB got the probable phosphoserine phosphatase, but mRub 0125, it got a protein with an unknown function. This is a weird result, but it doesn't confirm our argument, and it does not refute it either. It's just a weird result. After all of that, the two genes are predicted to have the same EC number, which is 3133, which is phosphoserine phosphatase. Nothing on this table contradicts that mRub0125 is actually phosphoserine phosphatase. In conclusion, we had a strong E value of the initial blast, same cellular location when the cytoplasm. They both pulled the same COG and PFAM hits. The PDB results were different, but it doesn't help or hurt us. The predicted function in the same KEG map, which is serine biosynthesis. Both were predicted to have the same EC number. They had similar conserved amino acids in the PFAM conserved sequence, and neither are a member of an operon. Our last comparison was between mRub 2910 and E. coli glyA. We once again started with our initial NCBI blast. The query sequence was mRub 2910, and we blasted against the amino acid sequence of E. coli glyA as the subject sequence. We can see a lot of similarities here. The E value was very low. We got 1 to the negative 154, which is super low and awesome. The bit score was actually pretty high for this one. All of this indicates a very strong sequence similarity. This slide is the results of the other bioinformatics tools that we used to compare, glyA and mRub 2910. In addition to the strong initial blasts, we see that they got the same cog number, which is cog 0112, which is glyA. They both didn't pull any Tiger Fam hits, which is weird, but since they both didn't pull anything, it's alright. They both got the same PFAM hit, which is PF00464, and all of them had very low E values. The protein database again pulled two different numbers, but as you can see, they both have serine hydroxymethyltransferase right here and here, and we can justify that they pulled them from two different organisms because E. coli and mRuber are not closely related. And they're both also predicted to have the same EC number, which is 2121, which is glycine hydroxymethyltransferase. In conclusion, they had a strong E value for the blast, same cellular location in the cytoplasm. Both proteins pull the same strong COG and PFAM hits. Neither of them pulled anything from the Tiger Fam database, which doesn't really help or hurt us. It's a little bit better that neither of them pulled anything rather than one pulling something. The protein database results were different, but again, it indicates that the same enzyme just pulled from different organisms. Both are predicted to be in the glycine biosynthesis degradation KEGG map, and they're both predicted to have the same EC number and have similar conserved amino acids when we compare against the PFAM conserved sequence. Neither of them are a member of an operon, which all of this concludes to their orthologous. So our final conclusion from this whole study has been that these bioinformatics tools help us to show that mRub 0173 and serA, mRub 0125 and serB, and mRub 2910 and glyA, they all show similarities in sequence, cellular location, and structure. So, overall, we can conclude that these pairs are orthologs. Thank you.